Warm greetings to my dear students. In earlier session we discussed about factors affecting enzyme action, and now we'll study about mode of enzyme action. So, which means how does the enzyme work? The working mode of all enzymes depend upon the nature of enzyme and the substrate which are participating in that process. So, enzymatic activity depends upon formation of ES complex, and for that process lowering of activation energy is required. Now, what is activation energy? So, energy required by the reactants to undergo the reaction is known as activation energy. So, mode of enzyme action involves two steps. First is formation of enzyme substrate complex, and second is lowering of activation energy. So, first step is formation of enzyme substrate complex. Let's look at the diagram first. See, this is enzyme molecule, this is substrate. The substrate occupies the active site and forms enzyme substrate complex, which further dissociates to form a final product. An enzyme is again free to react. So, Michaelis-Menten explained the mechanism of action of enzymes, and substrate must combine with the enzyme E combines with substrate S to form ES complex, which further dissociates to form enzyme and the final product. The final product is formed and enzyme is again free. So, Stern proposed that ES complex is an intermediate compound which is unstable. That means that it decomposes rapidly into enzyme and product molecule. So the mechanism of formation of ES complex can be explained by two theories. First, lock and key theory, and second is induced fit theory. So they both are very important from the examination point of view. First is lock and key theory. This theory was postulated by Emil Fischer in 1984. See, let's look at the diagram. This is enzyme molecule, this is substrate, and the substrate occupies the active site and forms the enzyme substrate complex. So in lock and key theory, the active site is pre-shaped and rigid. So the substrate fits into it. So it has a fixed template. That is why it is also known as Fischer's template theory. So according to this theory, the structure of enzyme is rigid. Like one lock has only one key. Similarly, a specific substrate only can bind with the specific active site of the enzyme. So now, next is induced fit theory. So according to this theory, first of all, this theory was postulated by D. Koshland in 1956. According to this theory, active site found is not rigid and not pre-shaped. As in lock and key theory, it was rigid and pre-shaped. So according to this theory, the active site found in enzyme are not rigid and pre-shaped, but as soon as the substrate comes in contact with enzyme, the active site changes and becomes according to it. So active site undergoes a minor change to bind with it and ES complex is formed. Let's look at the diagram. This is enzyme molecule. It has no rigid and pre-shaped active site. The substrate comes in contact with it and the active site brings minor changes in it and the substrate molecule fits into it. So this is induced fit theory. Actually what happens, you all know that enzymes are made of amino acids basically. So the amino acids, they are repositioned to form active site and bring about catalysis. So it was a more acceptable and realistic model. Now comes second step, that is lowering of activation energy. So now we know what is activation energy. The energy required by the reactants to start a reaction is the activation energy, and the enzymes, they have the property to lower down the activation energy. So when the reactants are heated, they achieve activation energy, they attain activation energy, and the enzyme, it reduces the activation energy and causes the reaction to proceed at a lower temperature. So the enzymes reduces the activation energy in such a way that all the biological systems occur at body temperature. See, this is the graph. The energy is plotted against rate of reaction. This was the activation energy without enzyme, and this is the activation energy with enzyme. So you can see the difference. That's all for now. We'll continue with the next topic, nomenclature of enzymes, in our next session. Thank you so much.